Hello everyone, my name is Bhavya Poliwal and I'm a master's student in the Department of Molecular Biology and Human Genetics. I began my program in the year 1142. Today I'll be presenting my progress report on my research topic identification of long non-coding RNAs in zebrafish glucocorticoid induced osteoporosis model under the guidance of my advisor Dr. Ming Dilling. My progress report has been reviewed and signed by my advisor. During this semester, I enrolled in a total of 7 credits. These included 2 required courses and 2 elective courses. The timeline of my activities include my arrival at the Suchi University on the 2nd of April 2026, and since then I have worked in my lab with paramecium filtration practice, literature review study, fish feeding, setup mating practice, pipetting practice, fluorescence image taking practice, and research topic refinement. In addition to my coursework, I have focused on gaining essential hands-on laboratory experience. This includes fish feeding, mastering pipetting techniques, and learning how to set up mating for zebrafish. I have learned the specifics of zebrafish husbandry, which involves feeding the fish 5 times per day with a variety of food, including fish food, paramecium for larvae, and preparing shrimp. I also manage the paramecium culture by feeding it yeast. Before setup mating, I distinguish males, which are typically brighter in color with slender bodies, from females, which are darker in color with rounded bellies. I have also spent time studying the zebrafish embryonic developmental stages. I have observed and documented various phases starting from one cell stage at 0.2 hours post fertilization through the 64 cell stage at 2 hours, the shield stage at 6 hours, and the 6 somite stage at 12 hours. By 72 hours, the embryo reaches the protruding mouth stage. To support the larvae, I practiced paramecium filtration and culture. Paramecium serves as a primary food source for zebrafish larvae. I have learned to maintain these cultures over a period of 2 weeks and monitor them using the compound microscope. I have practiced both standard and fluorescence picture taking practice to document my findings. My practice involved observing larvae at 8 days post fertilization, which is 8 dpf. I compared a control group kept in E3 buffer against groups treated with dexamethasone at concentrations of 2 micromolar and 4 micromolar to observe the effects of glucocorticoids. Turning to my research project is titled Identification of LNC RNAs in a zebrafish glucocorticoid-induced osteoporosis GIOP model. Osteoporosis is a condition characterized by reduced bone mass. In a healthy stage, there is a balance between bone formation by osteoblast and bone reabsorption by osteoclast. Osteoporosis occurs when this balance is disrupted, leading to increased bone reabsorption and decreased bone formation, which results in decreased bone strength and increased fragility. It is well documented that long-term glucocorticoid treatment can induce this condition. The zebrafish is an excellent vertebrate model for studying bone diseases for several reasons. Notably, approximately 70% of human genes have at least one zebrafish ortholog. Furthermore, their transparent larvae allow for the direct non-invasive observation of the skeletal development. My research focuses specifically on long non-coding RNAs, which are called LNC RNAs, which are molecules longer than 200 nucleotides that do not code any protein. LNC RNAs are vital because they regulate gene expression through several mechanisms, including chromatin modification, transcriptional regulation, and post-transcriptional regulation. They can act as guides, scaffold, signals, or decoy to influence gene activity. While there is an increasing evidence that LNC RNAs regulate bone metabolism, their specific roles in the GIOP model have not yet been comprehensively characterized in zebrafish. My experimental workflow will include zebrafish model husbandry, treating it with dexamethasone, which is a glucocorticoid, and doing bioinformatics analysis using a pipeline, and then functionally validating novel LNC RNAs. LNCR Analyzer pipeline is a publicly available pipeline. The first step includes raw RNA sequencing collection. The next is quality trimming to remove low quality bases. The third step includes reference guided transcript assembly using HiStat2 and string tie tools. The next is size selection based on LNC RNA size, which is greater than 200 nucleotides, and comparing the annotation with known RNAs. The next is coding potential estimation using several tools, and finally identifying novel LNC RNAs. My future directions will include GIOP model establishment and identifying novel LNC RNAs using the pipeline, then validating and functionally characterizing these LNC RNAs to better understand the regulatory mechanisms in glucocorticoid-induced osteoporosis model of zebrafish. Thank you for your time and attention.